Okay, good day, everyone, and welcome to the Marine Biodiversity Networking Friday session organized by the Earth Center. My name is Maria Gabriela Palomo, and today I have the pleasure to be the moderator of this session with Andrew Cooper and Bethany Reinhart, showcasing the Coastal Environmental Baseline Program, a Marine Life 2030 UN Ocean Decade Affiliate project. The program had the mission to better understand and manage Canada's coastal ecosystems and collaborate with different sectors to collect data on biological, biochemical, and physical variables. Let me introduce you now, our speakers. It is a pleasure to introduce Dr. Andrew Cooper. Dr. Andrew Cooper is a research scientist in coastal marine biodiversity with Fisheries and Oceans Canada at the St. Andrews Biological Station. He conducts research to measure and monitor changes in wild species diversity in the coastal zone to support regulations and conservation management objectives. He is the lead of the Mashpush Estuarine Marine Protected Area project and works with a broader team to identify and establish baseline biodiversity and habitat features for new protected areas in Canada. Andrew received his PhD in biology at the University of Ottawa with a focus on systematic zoology in marine species. The other speaker today is Bethany Reinhart. Bethany has her master in biology and bachelor in environmental biology from the University of North New Brunswick. She's an aquatic scientist biologist with the Fisheries and Oceans Coastal Environmental Basin Program. She actively engaged with the community organizations across many sectors to achieve common research goals. Bethany leads several projects that generate baseline data on coastal areas vulnerable to human impact. Her main research interests are on contaminants and species diversity in aquatic science. And before starting, let me introduce myself. I am a marine ecologist scientist at the Natural History Museum of Argentina. My research focus is on coastal biodiversity and how human activities impact the coastal environment, especially rocky shores benthic species. Before starting, please, if you have any question, you can write it in the chat here, and I will read them at the end of the presentation. So welcome both, Andrew and Bethany, and as you do a common presentation, the floor is yours. Thank you.
Thank you very much for that introduction. And thank you for the opportunity to come speak today. Let me know if you can see that.
Yes.
Okay, great. I just, before we get into the talk, I'd like to take a minute to acknowledge that the work that is carried out by the St. Andrews Biological Station in New Brunswick, Canada, where Andrew and I are both based out of. It takes place on the unceded territory of the Beniscoto-Mugati, the Wolastoqey, and the Mi'kmaq peoples, governed by peace and friendship treaties. And we're really fortunate to be able to get to experience this beautiful territory from both the land and from the ocean, and to build relationships with organizations that represent these communities in generating data together that helps us all better understand and protect the marine environment. So just to keep us on track, our outline of today's talk, I'm going to start and I'm going to give us an overview of the Department of Fisheries and Oceans Canada's Coastal Environmental Baseline Program. And then I'll talk about the local Maritimes chapter that we have in Atlantic Canada for this baseline program. And then Andrew's going to take over and chat about the MBON, Marine Biodiversity Observation Network, pole-to-pole rocky shore and sandy beach data collection that we do under the Coastal Environmental Baseline Program. And he's going to talk about how we chose our sites for the sampling that we do on rocky shores and sandy beaches, what we've found so far, and then the future plans that we have for the MBON work that we're doing. The Coastal Environmental Baseline Program is a Department of Fisheries and Oceans Canada program, and it was established in 2017. It was a number of initiatives that were created under the Oceans Protection Plan, and it was under the pillar of protecting the marine environment. The objective of the program, one of them is to collect baseline data to allow the characterization of the current state of key coastal ecosystems. And we identify those as being subject to high vessel traffic or increasing vessel traffic or coastal development. We want to collect this baseline data in a way that is in sufficient enough detail that will allow us to detect future changes, detect changes over time, and provide high quality data as well that can be used to inform decision making. Something of note is that the program has also been endorsed by the UN Decade of Ocean Science, and so it's an affiliated project of the Marine Life 2030 program. The baseline program uses a coordinated national approach, which is based in Ottawa, and it's implemented at the regional level by DFO scientists across 6 different regions across Canada. The program is also implemented in a way that emphasizes engagement. Engagement is a huge part of what we do within this program. So we work closely with program participants to design together different aspects of the program and identify data collection priorities. So what are we collecting data on across the country to create these baselines? So asking the people that live and work and study in a specific area, what data is a priority for you and this area that you're living in, and then enabling these people to also carry out the data collection projects for themselves. Another big part of the program is to publish all of the data that's generated openly online. So our partners work with the St. Lawrence Global Observatory here in Canada, or our department's open data portal, or another equivalent open data platform to ensure that everything's meeting international standards before publication. So a set of nationally consistent target ecosystem components were identified as being the most important components to capture in these data collection activities. So these categories are the key physical and biological features that are needed to develop a baseline for these coastal areas. And they're also features that all of our partners care about and are most interested in collecting for their specific local areas. We've now moved towards aligning the program's target ecosystem components with essential ocean variables and essential biodiversity variables, EOVs and EBVs, to increase the interoperability and utility of the data for the future as well. So this awesome graphic summarizes some of the phase 1 successes of the baseline program. There were 6 original study areas from across Canada identified here on the map. So from west to east, we have the Port of Prince Rupert in British Columbia, the Port of Vancouver in British Columbia, Iqaluit and Nunavut, the St. Lawrence Estuary in Quebec, the Port of St. John in New Brunswick, which is the Maritimes chapter, our Atlantic Canada chapter for that Andrew and I are situated and kind of did this work under that chapter of the program, and then Placentia Bay in Newfoundland and Labrador. Our team from the Maritimes region was working out of the Port of St. John from 2017 until 2023. And the main takeaways here are that the bulk of the data collection projects that the baseline program funded in this time were led by nonprofit organizations, Indigenous organizations, and academic institutions. So partnering organizations. And then there were 98 projects that were led by these partners, and then 61 projects led by DFO, fisheries and ocean scientists as well. This resulted in over 80 openly available data sets. So a ton of data is being generated across the country on a lot of different ecological topics, all with the intention to be shared openly online. So it's a huge effort, and our Maritimes chapter is just one component of this much larger effort. And so now, hopefully, I'm going to share with you a video that was developed by our national team for the program. We had a workshop in Ottawa where DFO scientists and all of our participating organizations were invited, and that's where a lot of these interviews took place. So I think it's really nice to hear about the program from the perspective of the partners. So hopefully this works.
The Coastal Environmental Baseline Program collects baseline ecological data at 6 key coastal sites in Canada. What's unique about the approach that the program uses is that Fisheries and Oceans Canada works with partners to work on different science projects to collect all the data that's needed to establish a baseline.
Canada is a huge country with a very long coastline, so supporting the people who live in these regions to describe the ecosystems is a really important win.
It gives us the possibility to have the tools to better manage our regions and expand our knowledge.
With First Nations, we are the primary people that are going to notice something is going off because we're always out there practicing our traditional rights.
We're here 365 days a year. We know so much that DFO can use.
I think it's really great to be asked what's important to us and to take the knowledge of all the different partners on board in structuring the program.
As a small NGO, we've been able to employ multiple people to work on these projects and building that capacity within our local region to support more research going forward.
This program helped to fill data gaps, habitat, species population, species at risk in the St. John Harbour.
Our niche is diving, so we can share all of the data we've collected with the public. It's very interesting.
The way DFO has been doing their program up here, asking the community what we want, are interested, asking the community what we know is going on, and asking how they can help us expand our knowledge into two fronts, the traditional and the scientific knowledge. I find that, that's probably the best approach.
It's been really rewarding to be able to work so closely with Indigenous organizations and nonprofit groups and the fishing industry and academics. Everyone brings their own ideas, and they've been collaborating on new studies for the future as well.
We are currently in a historical global moment where change as a result of the changing climate is inevitable.
What we do is important for Canadians because we are trying to understand the environment that we all live in.
There's so much life at the bottom of the ocean that contributes to the functioning of the system and all the benefits that we derive from that. So understanding about the distribution of the animals on the seafloor is crucial for that.
20, 30 years in the future, we need to have this information in order to be able to determine how things have changed and how things are going to continue to change.
The work that our organization did in concert with countless other organizations will provide a baseline for decision making, potentially for the next decades, for policymakers and for really everyone in the country.
Now that I find myself as the next generation of Inuit men who are going to be helping in this kind of research while also taking part in our traditional activities, it's a really great feeling. And I hope it continues on into the future for the next generations.
So that was the program as a whole, a picture of the program as a whole. But I wanted to also give you a quick and broad look at what we were doing in the Maritimes region specifically. So there were 29 projects that took place between 2017 and 2023. And we have 15 additional projects underway right now. And in our region in Canada, we've had 14 organizations that have participated and received funding to lead these data collection projects that span a wide range of subject matter. So on everything from microalgae to seal populations, physical oceanography, surface currents, to model where oil spills may travel. And so I created this word cloud using our regional priorities that each of our phase 1 funded projects sort of touched on within the St. John Harbour. So in a word cloud, the bigger the word is, the more often it shows up within the submitted text. So you can see within the projects, the priorities that were being addressed. Biodiversity is there in very big font, as well as species and contaminants. So again, these are the priorities that were and the projects that were formed after going to these community organizations, Indigenous organizations, nonprofits, academics, and asking them, what do you guys care about for the St. John Harbour specifically? And, it's a busy port area. Andrew's going to talk a bit more in a few minutes about the Port of St. John. So I won't get into that, but it's no shock with the area that biodiversity and contaminants are some of the most large priorities for the area. Our internal team at DFO for the baseline program has led data collection projects as well. So our projects mostly focus on physical oceanography, characterizing substrates, also contaminants analysis, and then biodiversity. And so this is a great point for me to pass the mic over to Andrew, who's going to talk about the work that he advised us on and that we co-led on looking at intertidal biodiversity through MBON, Marine Biodiversity Observation Network, pole-to-pole protocols that we carried out for sandy beaches and rocky shores within the St. John Harbour and the Musquash Marine Protected Area.
Oh, okay. Hi, everyone. Thank you, Bethany. Thank you to everyone who is online today. Bethany, I'll get you to change the slides for me. I mean, I'm not even sure anybody can see me, but as long as everybody can hear me. Yeah, as Bethany indicated, there are many projects conducted in and around the St. John Harbour area during phase 1 of the baseline program. I'm just going to take a few minutes to give you an overview of just one that used the MBOL and pole-to-pole, rocky shore, and sandy beach protocols. We conducted this work at 2 locations close to the St. John Harbour. One was at a Mispec Beach location, which is just a few kilometers to the east of St. John Harbour, the busy harbour of St. John. And the other was at Black Beach, which is about 20 kilometers to the west of St. John Harbour, and conveniently within a federally designated marine protected area. Next slide, please. So just a summary of the site characteristics at these locations. The Mispec Beach location had both sandy beach and rocky shore intertidal habitat. The sandy beach, which you can see in the upper photo, is quite close to the Port of St. John. There's a Cannon Port liquid natural gas port terminal right in the background. It was a fairly large dissipative lacustrine source beach, about 600 meters in length. Had a fairly large mesolittoral zone. So that's that zone, that intertidal zone that gets exposed between the high and low tides. It was about 100, we estimate about 100 meters. It's a southwest exposure, so it does get some prevailing weather, but typically we're noticing that the wave heights in here is less than a meter. The rocky shore, which was adjacent to the sandy beach, and in the upper photograph, you can kind of see it in the distance, the dark area in the distance upper left. Again, this is and then the close-up photo in the bottom there. It was characterized by large boulders and bedrocks. It was about 50 meters in length. That mid-tide zone, that mesolittoral zone, was about 20 meters, and it had a northerly exposure. So not getting a lot of waves, more waves are coming in sort of on the side. It'd actually be if the waves were bigger in this area, it'd be a great shore break for surfing. Next slide, please. The Black Beach location also conveniently had the sandy beach habitat and the rocky shore habitats in close proximity to each other. Again, the upper photo is Black Beach. It's called Black Beach because the sand is sourced from graphite. It's not volcanic like other black beaches in the world. It's a graphite source. It's another dissipative lacustrine beach, about 200 meters in length, a large mesolittoral zone. And the reason why these mesolittoral zones are so large or relative to the size of the beach is because we get tidal amplitudes from 6 to 8 meters in this area, so which is quite a lot of tide. We have a western exposure on this beach, but it's quite protected as it's situated inside the harbor, and typically wavelengths are less than half a meter, or typical wave heights are less than half a meter. The rocky shore for this is again seen in the background on the upper photo and a close-up in the lower photo. You can see that it's a dense coverage of Ascophyllum nodosum, the brown algae. Its features are bedrock with large fissures. It was about 150 meters in length, about a 30-meter mesolittoral zone, and again, western exposure, but quite protected because of that harbor situation. Next slide, please. So just an aside on these site characteristics compared to what the MBON protocols identify as preferable beach habitats, and this is mostly for the beaches. So the MBON protocols really are looking for sites that are oceanic exposed and avoiding embayment in closed beaches. And we are noting here that neither of our locations, the beaches are oceanic and exposed. The upper photo, again, is another angle of that Black Beach in the marine protected area. You can see that it's within a harbor and there's a shoreline on the far side, so it's not very exposed. What we're finding that the beaches that are oceanic and exposed have a lot of energy in them, and they're really quite steep. So the lower photo shows typical exposed beach from this location, from this region, and quite, we get large boulders, very, very steep, not very dissipative, and almost impossible to sample with the protocols that are in the MBON pull-to-pool methodologies. We discussed this for some time as to trying to choose the best locations, and probably agonized a little bit over it, but despite the shortcomings of these beaches, we felt that these two locations really best suited the needs of the baseline program and the MPA. And I'll explain that in the next two slides. Next slide, please. So the Mispec Beach location was, as I mentioned, was really close to St. John Harbor. It's a major marine port with high shipping traffic. It's an urban center. There's dredging, there's discharges from municipal wastewater, industrial effluents. There's an oil refinery, brewing, pulp and paper production. And so the level of activity in the St. John Harbor makes the local marine system and surrounding shorelines vulnerable to cumulative impacts and environmental change where we needed baseline information. And that Mispec Beach location was suited those needs. Next slide, please. For similar reasons, the Black Beach location was also within the study area for the baseline program, but as I mentioned, designated as a national marine protected area, and it was very important for fish, birds, invertebrates, and marine plants. It's the largest ecologically intact estuary in the Bay of Fundy. It has many distinct habitats within a fairly small 7 square kilometer footprint. The monitoring plan that we have in place right now is to establish baseline information for productivity, for biodiversity, and the different habitats. And so the MBON protocols were a very good fit for supporting the current monitoring plan for this MPA, because we had a lot of gaps in information, particularly in those intertidal habitat, highly with a lot of biodiversity. Next slide, please. So just to get into some details with the sampling and our data, this is a close-up picture of our Rocky Shore site. In the upper right, you can see that it's that dense coverage of Ascophyllum, that brown algae. It's typical for this region, and that's typically the canopy, the upper canopy for most of our rocky shores. But when we move this canopy aside, it really reveals quite a fantastic community of barnacles, periwinkles, and other algae. All are to be identified and counted by our sampling teams, and you can see them here with the standardized transects and that were created through the MBON pole-to-pole protocols and including the camera transects. As per the protocols, we split the beach into 3 strata. The swash zone, which is that low tide area, the mesolittoral, which is that mid tide area that gets exposed, and the superlittoral, which is our high tide zone. And within each of those zones, we do 10 quadrats, 50 centimeters by 50 centimeters. So the total of 30 quadrats per site. During the day, we do the sampling in 1 low tide cycle, 1 day, so 1 site per day. We identify everything within the quadrat, including the percent coverage of the canopy layer, and identify and count all the invertebrates. Some of the samples are taken back to lab to confirm identifications, but in general, at the end of the day, we have all the data in hand, and everyone seems to be smiling because they've just spent a great day on the rocky shores finding all these little critters. Next slide, please. Just look at our preliminary results. This is an OBIS generated observation summary broken down by larger taxonomic units, and it's for our results for years 2021 and 2022 for all the rocky shore. You can see that the observations are dominated by that Phaeophyceae class, which is brown for us, brown algae, mostly species of rockweed, that Ascophyllum podosum, but we also have 3 species of fucus, the bladderwracks, and species of edible dulse and kelps in the low intertidal area. And these contribute a significant proportion of the canopy, that secondary layer. Pulling back that secondary layer, looking at the primary layer, we've got a lot of multicellular red algae, that Floridophyceae, and as well gastropods, periwinkles, amphipods with the occasional green crab, and barnacles, the Thecostraca. So we took some time. Oh, next slide, please. So we took some time to try to understand the variability in our data so far, and we realized it's still early days in understanding what we have. This plot you're seeing here is a non-metric multi-dimensional scaling using a Bray-Curtis similarity matrix for both Mistback Beach and the Black Beach locations for rocky shores, and both years combined. The colored clusters are bootstrap averages for each strata within each location, and then the big circles are just hand-drawn around the locations just to help visualize the 2 different locations that we're comparing. So what we're finding is that there's a difference between the 2 locations, as well as there are clear differences among the different strata within each location. The most common species observed across all these sites were the ones you might recognize as very characteristic of our shorelines, of course, that Ascophyllum, Hildebrandia, common periwinkles and barnacles. We can see the differences between the 2 sites, but we can also see, particularly for Mistback Beach, a strong gradient between high on the left to the low tide area on the right, like a continuum. And it's really changes in the Ascophyllum community, changing from abundances of Ascophyllum, fucus, and associated species. Next slide, please. So we can see that we go back to observations, photos of the area, and we can actually physically see that on the rocky shore. And this is just a photo of the high tide, that transition between high tide and mid tide zone. I'm not sure how clear it is to people on Zoom here, but if you look carefully, you can see that transition between that knotted rack, that Ascophyllum on the right in the lower tide area, that mid tide area, up to that high tide where you see a lot of bladder rack type of fucus species. And that was a very telling and sort of affirmed what we were seeing in our data analysis to date. Next slide, please. So for the sandy beach sampling, the MBON protocols had us split the beach into 3 strata. This time we take core samples of the sand. There were 4 core samples per strata, so we end up with 12 core samples at each beach. It doesn't seem like a lot, but we realize that's a lot of sand to then sieve through and tease out the organisms that are in that sand. So we decided that on site we would do an elutriation process. And this is a very simple suspension of the core sample with seawater. We do that several times to help separate the organisms into a light component and the heavy sediment into a heavy component on the bottom. For the first 2 years, we did a lot of comparison of both our light and heavy samples to assess the optimal number of times that we should elutriate and how much information we could be losing by disposing of the heavy sample and just analyzing the light sample. And we estimate we're about 90% effective, but this is a huge time saver. And we figure that losing, less than 10% of the information is, as long as we're consistent with this, that it's a reasonable approach for dealing with this much sand. The samples are taken back to lab. They're preserved and isolated from the remaining sediments, and then we send them off. We currently are sending them off for identification at the Atlantic Reference Center because we don't have a lot of in-house taxonomic expertise for these groups. I'd like to mention that for both Rocky Shore and Sandy Beach, we record other information as well, like shoreline rugosity, slopes, wave characteristics, temperature, and grain size. Next slide, please. So another OBIS-generated observation, a taxonomic summary of the data for the Sandy Beach. And within the Sandy Beach samples, the species assemblage was dominated by copepods, Calanus species, polychaetes, and also a pretty good proportion of annelid worms, the Clitellata. There was a lot of juvenile stages in the sample and a lot of very important forage species. So it really tells us a lot more information about what would normally look like just a sandy beach, of actually the diversity and the importance of sandy beaches for these organisms. Next slide, please. So again, preliminary results. There were 32 species observed at Black Beach and 30 species at Mispec Beach over those 2 years. Again, significant differences between the 2 locations and significant differences among the strata within each of those locations. One interesting note is that although the 2 locations had approximately the same number of species, only 9 of those species were actually common. The others were all unique to the location, which is kind of an interesting aspect of the diversity and the changes from 2 locations that are geographically fairly close to each other, but perhaps have slightly different geologies and characteristics. So at this stage, I'm going to hand this back to Bethany to provide some summary comments, but obviously thank you and happy to answer questions that you may have at the end.
Thanks, Andrew. So that's the content that we have to present today. So in summary, the Department of Fisheries and Oceans Canada's Maritime's Coastal Environmental Baseline Program and the Musquash Marine Protected Area programs give us an opportunity to apply these MBON pole-to-pole sampling protocols to these standardized global protocols to vulnerable habitats in the region. These programs that we have based in Atlantic Canada, they share similar objectives in generating baseline data in vulnerable areas like the Port of St. John and the Musquash Marine Protected Area, working with community organizations to generate this data as well. So it really made sense for us to work together and align our goals. We faced challenges with finding best practice locations. So the data we've generated may not be directly comparable for ideal MBON locations as defined within the protocols, but Mispec Beach and Black Beach locations serve our program objectives pretty well. And of course, we're in the preliminary stages to fully describing the variability within our data, but we have learned to successfully apply these MBON protocols, publish the data to OBIS, and have started to gain a much better understanding of the diversity of species in these habitats as well. And we're also working on future plans for the program as well. So our next steps with our MBON data collection are to take a deeper dive into the data and look at the specific differences that we're seeing across years as we generate more years of data, and look at the differences across sites and strata, and compared to historic data that may have been collected in the region. And there's environmental data, Andrew mentioned, that we can incorporate to interpret, help us interpret the differences that we are seeing across sites and across strata. The benefit also of this being standardized protocol is that, there's other researchers using MBON, pole-to-pole, rocky shore, and sandy beach protocols. So I know there's some data that's based out of Maine in the United States, right next door to us. So it will be interesting to look at their biological communities as well, and do a comparison. So we're going to continue the sampling program for MBON, and we'll do that at the 2 beaches that Andrew mentioned. And we've also added a couple other sites going west within the Bay of Fundy coastline as well. We're going to continue to publish our data on OBIS from 2023 onward. And we also had the opportunity through the work we've done so far to take out 2 baseline partnering Indigenous organizations and show them how we carry out these MBON protocols. And we're very happy to continue to do this. The connections that we have made through the National Baseline Program could help MBON pole-to-pole sampling efforts to kind of spread further across Canada. So I think that's great. This is our MBON sampling team. It's a lot of work and very fun work. So thanks to everyone that helped us along the way, and a special thanks to the Passamaquoddy Recognition Group and AMIC, who came out and learned the MBON protocols with us. And that's all from us. Thank you very much for listening. And thank you for the invitation to come speak again.
Thank you very much, Andrew and Bethany. Very interesting and a lot of work. I'm sure it is a hard work in their latitudes to work on that side. So we have some questions, and Nidhi Gopal asked, is there any research on phytoplankton and how leaf epiphytes, microalgae, and bacteria affect the seagrass ecosystem function as greenhouse gas sink and the greenhouse gas production under environmental stress? If yes, kindly explain. Thank you.
Just reading.
Yeah, Bethany, I said I'd answer all the questions, but I don't. Within the baseline program, I don't know. Are we doing Are they doing some phytoplankton work in the area?
There is a researcher, so Dr. Gary Saunders from the University of New Brunswick leads a project looking at microalgae communities, and it's a lot of what he's doing is sequencing. So that I think there's some metabarcoding techniques being applied to create a baseline inventory of the microalgae communities here. So under the baseline program umbrella, that is the extent of the microalgae work. And it's not my area, so I can't speak to any other microalgae and bacteria work that's being done elsewhere, but I do know that, yes, we have funded some microalgae content.
Okay. The second question, how are the local communities and their traditional knowledge incorporated in the DFO baseline projects?
That's a great, so in the beginning phases of the program, which I was not present for, and Britt Dupuis is on the line, feel free to jump in if you have any input, but so there was a lot of engagement within with the local regional aspects. So the leads within the region go to the community organizations and Indigenous organizations, making it a priority to gain their input on those priorities from the onset of the program, to say, you know, what do you want to see collected under this umbrella? And so a lot of that feedback, and then there was workshops to develop project proposals and develop the actual data collection as well. And we're consistently, you know, in touch with Indigenous organizations within each region to make sure that their input is incorporated into the design and the development of all of these projects in an ongoing way too.
Okay, thank you. And you mentioned that some of the data was available through the DFO open portal. Is there a DFO open portal?
Yeah, there's a DFO open data portal. So all of the fisheries and oceans scientists data will eventually be online within the open data portal, or on OBIS, or another equivalent open data portal. But yes, there is, I think it's called Open Data, but there's a Fisheries and Oceans Canada open data portal.
Yeah, and just to add that the data that we're showing today has all been published on OBIS.
Oh, good. And next question: how do you see the incorporation of the EDNIA analysis of and remote sensing to characterize biodiversity and regional scales?
Could you repeat the question? I didn't.
Yes, it was moving all the time.
Yeah.
How do you see the incorporation of EDNIA analysis and remote sensing to characterize diversity and regional scales?
Yeah, so we do have some, if I can speak to that, we do have some projects looking at eDNA within the Musquash Marine Protected Area and the surrounding coastlines. And that's sort of like a new field for fisheries and oceans to incorporate into just understanding coastal biodiversity and monitoring itself. So there is some early work being done with eDNA, and the rest. And yeah, not aware of the rest. Whether there's a lot being done with water quality in the area. It's quite a dynamic coastline, so as we're finding with the biodiversity in terms of sampling, that type of thing, it's quite intensive. So there's a lot of changes even from embayment to embayment. So the sampling, trying to understand what's happening broadly on the coastal side with that much tidal energy is certainly complicated and requires quite a lot of effort. So
yeah. Yeah, but it is not part of MBON protocol, is it?
No, it's not. No, that's correct. Thank you. Yeah.
Thank you. Next question: which university have you mentioned, Bethany? Madam, can you repeat it, please? Okay.
Yeah, that was I was just going to type it out, but that so that was Dr. Gary Saunders that had done the microalgae and the algal inventory work under the baseline program from the University of New Brunswick in Canada. And his data should be online on the St. Lawrence Global Observatory platform.
Okay. And next question: will regulatory agencies provide guidance on the application of this type of method to be able to scale up biodiversity analysis? Yeah. Is there any? Yeah.
It's like you're cutting out almost. Could you repeat that?
Yes, if there is any agency that support the MBON protocol research?
Yeah, so within Canada, no, we were probably, DFO is the first to establish sites within Canada. I think the baseline program is now one of our opportunities to communicate this to other researchers, other interest groups, to sort of educate them for those protocols, and see if they have any interest in adopting that type of sampling. So at this stage, yeah, the first two sites in Canada were those two sites, the Spec Beach location and the Black Beach. And now I think part of our future plans is just try to communicate how to use those protocols here and that they're available. And if people are interested in that intertidal biodiversity work, that you could apply those protocols.
Thank you. And I have a question. Monitor of biodiversity in coastal areas are very important, and especially if we can keep the same protocol along all America, it's really a very good baseline for management decisions. But what do you think of looking to the future? What do you think it's a good advice to, what we need to change as populations, as humans, to protect and to conserve these environments, especially rocky shores?
Well, good question. Yeah, so certainly your point about having standardized protocols to be able to compare changes in biodiversity on a global scale is really important. And it's one of the reasons we agonized a little bit by diverging a little bit from that Sandy Beach ideal location. And to use the results from that to inform on human use activities and things that could change. At this stage, my feeling is that, just understanding what is there. Like, you look at a beach and all you see is sand. And then to understand that it's a really important habitat for early life history stages, essential forage species, that it's not just an empty sand beach. And to be able to communicate that to the public, to people that are in charge of managing these environments, I think is key. So I think just even if we don't have the answers of what you need to change, understanding the importance of these habitats in the broader ecosystem is really key.
Okay, thank you. Well, we have two more last minute questions. The first one is, are you picking up a lot of invasive species?
Certainly it's on our radar, the invasive species. For the rocky shore areas, yes, the green crab, we're looking out for other crab invasive species. There's a number of bryozoan species that are maybe invasive on kelp that we're taking a look at, as well as some red algae. I don't have specifics particularly on the movement of some red algae species, but it's certainly the protocols that are in place. If we continue the monitoring, we should be picking up the presence or changes in invasive species.
Okay.
Good question, thank you.
Thank you. The Marshwash MPA's conservation objectives focus more on ecosystem structure and function. From the data generated through MBON and CEBP, do these more general conservation objectives resonate with findings? Rather than having MPA design and management tailored to protecting one or two keystone sentinel species?
Yeah, a really good question, and obviously something we have to think about in terms of moving forward with monitoring biodiversity is getting an overall picture of what's there, and then do you focus on, once you've discovered the diversity and species that are actually there, then move forward to say, well, we're just going to focus on key species and changes, whether it's an invasive or a species at risk. But I think as a baseline, you have to sort of go in and identify everything and not assume that, we think that the species is there and that's all we're going to focus on. You really do need to understand the larger picture first and then move forward from that.
Okay. And the last one, are there any existing global data repositories for eDNA data yet? Well, yeah.
That's a little, it's a little out of my field. But with the eDNA work that we're doing, we do, again, the data that we are collecting is going to be openly shared, and there are, repositories of molecular information libraries, eDNA, like metabarcoding libraries that are used for comparison of the eDNA that we're sampling to what's actually in the library. I'm not sure that answers the question, but that's sort of, there are global repositories for molecular biology, like sequences. But in terms of eDNA itself, I think at this stage there is some development for common eDNA discovery libraries, but currently we're just going through our open source, open government data avenues of reporting of what we find.
Okay. Thank you. That's all the questions for this session. And thank you, Andrew and Bethany. It is a very, very nice presentation. I think it is very important to monitor what is happening there, and the M1 protocol is a really good way to estimate biodiversity in rocky shores or sandy beaches. And yeah, and I think it's a long way to go. We need to still work on more stuff, especially in for conservation management, especially in this changing world. So thank you very much, and I hope you enjoy the session. Thanks.
Yeah. Again, thank you for inviting us. It's a pleasure.
Pleasure. Bye.
Thank you. Bye.
Thank